We are all taught the same formula in school. XX means you're a girl, and XY means you're a boy. But for many of us, especially if you're trans or gender nonconforming, that story has always felt incomplete. What if I told you XXXY is just the first sentence of a 7-chapter biological epic? That simple pair of chromosomes is just the first domino in a fascinating, complex cascade that builds our physical sex characteristics. The real story is far more intricate than a couple of letters. It's a stunning biological process with multiple layers, and crucially, those layers don't always align in the way we're taught they should. Today, we're going to break down a helpful way to think about sex development in humans into layers. This isn't just a science lesson, it's a framework for understanding the beautiful complexity of our bodies and why the concept of sex assigned at birth is only a tiny part of the story. It all starts at fertilization. This is layer 1, chromosomal sex. Our DNA organized into 23 pairs of chromosomes. The 23rd pair is the sex chromosome. Typically, an egg carrying an X chromosome is fertilized by a sperm carrying either an X or a Y. This results in an XX or XY pairing. This is often called the starting instructions for a female or male body, respectively. But right away, we see this isn't a strict binary. Some people are born with different combinations, like XXY, Klinefelter syndrome, or a single X, Turner syndrome. These chromosomes are the initial instructions, but the final outcome depends on how these instructions are read and followed in all the layers to come. If chromosomes are the blueprint, what starts construction? That's layer 2, genetic sex. This layer hinges on a critical gene on the Y chromosome, called the SRY gene. Think of it as a master switch of sorts. Around week 6 of development, if the SRY gene is present and functional, it flips on and triggers a chain reaction, essentially telling the body, let's build a male. The SRY gene activates another gene, which gets to work building testes and actively suppressing the development of ovaries. If there is no Y chromosome, and therefore no SRY gene, a different set of genes take the lead to direct development along a female pathway. This is an equally active and complex process, not just a default. This layer shows us that it's the genes, not just the chromosomes, that are calling the shots. For example, a person can have XY chromosomes, but a missing or non-functional SRY gene. Without that switch, their body will develop female characteristics. Once those genetic switches are flipped, we hit layer 3, gonadal sex. For the first 6 weeks, every embryo has what are called bipotential gonads, internal structures that have the potential to become either testes or ovaries. In an XY embryo with a functional SRY gene, these bipotential gonads are instructed to develop into testes. These new testes are about to become tiny hormone factories that will direct the rest of development. In an XX embryo without the SRY signal, a different genetic pathway guides those same structures to become ovaries. This layer is critical because the gonads, testes or ovaries, are what will produce the hormones that physically sculpt the rest of the body. Variations at this stage, where there's a mismatch between chromosomes, genes, and gonads, are associated with many intersex variations, also known as difference of sex development. With the gonad factories built, we enter layer 4, hormonal sex. The new testes or ovaries get to work producing and releasing sex hormones, chemical messengers that guide the next phase of construction. In male development, the fetal testes produce two key things: testosterone and anti-Müllerian hormone, AMH. Testosterone begins masculinizing the body, while AMH has a specific demolition job we'll cover in the next layer. In female development, the key factor is the lower androgen levels, combined with active ovarian signaling. The fetal ovaries produce their own hormones, like estrogen, but it's the lack of the strong male hormonal signal that allows the body to continue on a female developmental pathway. But it's not just about what hormones you produce. It's about whether your body can listen. In androgen insensitivity syndrome, a person with XY chromosomes and internal testes produces testosterone, but their cells can't detect it. Because their body can't hear the testosterone, they develop female external features. Those hormonal signals now go to work on layer 5, internal and anatomical sex. Every fetus starts with two sets of internal plumbing: the Wolffian ducts, male precursors, and the Müllerian ducts, female precursors. In male development, the AMH tells the body to get rid of the Müllerian ducts. At the same time, testosterone stimulates the Wolffian ducts to develop into vas deferens and seminal vesicles. In female development, the opposite happens. With no AMH, the Müllerian ducts flourish and develop into fallopian tubes, uterus, and upper vagina. And with no testosterone, the Wolffian ducts simply fade away. By the end of this stage, the internal reproductive anatomy is set. While the internal plumbing is being sorted, those same hormones are shaping layer 6, external anatomical sex. Everyone starts from the same basic embryonic structures or materials: a genital tubercle, urogenital folds, and labioscrotal swellings. These structures are neutral, waiting for hormonal instructions. In male fetuses, a potent form of testosterone, called DHT, sculpts these structures. The genital tubercle becomes the penis, the folds fuse to become the urethra, and the swellings merge to become the scrotum. In female fetuses, the absence of DHT leads to a different result. The genital tubercle becomes the clitoris, the folds become the labia minora, and the swellings become the labia majora. The penis and clitoris, the scrotum and labia, they all develop from the same starting points, just guided by different hormonal signals. This is the layer that is used to assign sex at birth. Finally, we arrive at layer 7, puberty and brain development. After birth, a second massive wave of sex hormones arrives during adolescence. This hormonal flood drives the development of secondary sex characteristics. In females, estrogen leads to breast development and wider hips. In males, testosterone deepens the voice and causes facial hair to grow. This is the stage where the physical differences that started in the womb become more pronounced. But at the same time, these hormones are also acting on the brain. The research suggests prenatal hormones help shape aspects of brain development, and puberty continues the process. Critically, gender identity arises from the brain shaped by both biology and lived experiences. This is our innate internal sense of being male, female, both or neither. For many trans people, the hormonal shifts of puberty are what bring a mismatch to the surface. Your mind and internal sense of self feel one way, body is being pushed in a different direction. This final layer shows that sex development is a lifelong process, and it highlights the distinction between the body's development and the brain's identity. So there you have it, the 7 layers of biological sex: chromosomes, genes, gonads, hormones, internal anatomy, external anatomy, and finally, pubertal and brain development. What this journey shows is that biological sex isn't an on-off switch. It's a sequential, multi-step process. The sex a doctor assigns you at birth is based on just one of those layers, external anatomy. But as we've seen, any of these layers can vary. This biological complexity is not just an interesting fact, it's a powerful validation. It helps explain why someone's internal gender identity may not align with the sex they were assigned at birth. That simple XXXY story isn't just incomplete, it's misleading. The reality is far more nuanced and frankly far more beautiful. If this deep dive helped you understand yourself or the world a little better, hit the subscribe and let me know in the comments if any of these layers was a big aha moment for you. Thank you all for watching.